generalized additive modeling, generalized linear mix modeling, and maximum entropy methods to estimate the distribution of species. So these were standalone studies, but also contributed to the parameterization of Atlantis. Some of the innovations of these methods account for spatial autocorrelation and presence-only data, which is particularly advantageous for modeling the distribution of birds and mammals, which often rely on opportunistic reporting by citizen scientists. Another method that we've developed in support of ecosystem modeling is a statistical model for the description of diet composition. Predator diets are bootstrapped and fit to a Dirichlet function, which is the multinomial generalization of the beta function. This accounts for covariation between prey items and is less sensitive to rare feeding events than alternative methods that are typically used to parameterize a diet matrix. With the description of error, we can engage in Monte Carlo simulations of ecosystem models accounting for a major source of uncertainty in the diet matrix. Michelle Massey, my former PhD student, helped advance this method in her dissertation, and Becky Scott, who is now working on her PhD, is using a related approach, which I'll discuss in a moment. This is an overview of the data synthesis in Atlantis from the CIMAGE project, and I'll just highlight a few linkages. We use mesocosm experiments, which were done at Moat, and fish lesion data from long lines provided by Steve Murawski to parameterize a dose response model. It's integrated in Atlantis. Oil fate data from the University of Miami is employed, as well as data from high pressure biodegradation experiments, which were done in Germany. Toxicity is estimated from sediment cores, partly with data from Isabel Romero and David Hollander here at CMS. We incorporated invertebrate sampling from the Hart Institute at Texas A&M and ROV data from the University of Florida. So these are some of the results from the comparison of Deepwater Horizon and Ixtoc. It's noticeable, particularly with Deepwater Horizon, that the effects are translated far away from the initial spill locations due to impacts on larval transport and impacts on mobile forage fish populations. In the case of Ixtoc, the oil kind of moved through the system quickly to the northwest, while with Deepwater Horizon, the oil remained in the same place for a longer period of time. And I think that difference is responsible for some of the differences that we see between the spills. So these are biomass trajectories showing the initial impact from the oil spill and then recovery. The Deepwater Horizon is in black and Ixtoc is in gray. So in both cases, the Deepwater Horizon and Ixtoc showed a sharp decrease in abundance across a large number of species, but Deepwater Horizon showed a more monotonic recovery back up to the pre-spill condition, whereas Ixtoc showed a more chaotic recovery and indicated possible stable state changes. And I suggest that one reason for this could be that the Deepwater Horizon oil kind of stayed in one place and impacted everything strongly, so the whole ecosystem rebounded together. In contrast, the Ixtoc oil sort of rifled through the system, and so the timing of impact and recovery was not coordinated among species. In addition, there were major impacts on juvenile fish. This is mostly owing to the fact that they're located inshore where they were exposed for a longer period to the oil. So this has led to recurring recruitment lows that followed the initial impact, and these recruitment lows have persisted over several generations. So we have reason to believe that there were impacts to the mesopelagic fish community, and this can hold implications for exploited pelagic species that rely on mesopelagic forage. And so this is the subject of Becky Scott's dissertation. This is an echogram of the West Florida shelf taken from a CIMAGE cruise in 2018, provided by Steve Murawski. And this illustrates three mechanisms for exchange between the meso and epipelagic food webs. A shows small mesopelagic fish, mainly myctophids, undergoing diel migration to the surface at night and becoming prey for tuna and billfish. B shows large predators like swordfish and escalars migrating to the surface at night to feed. And C shows large pelagic predators which have been found with non-migrating mesopelagic prey in their stomachs, mainly hatchetfish, suggesting that they hunt in deep water. So that's at least three connections between epi and mesopelagic food webs, which Becky is representing in Atlantis. So obviously, to simulate the effects of the pelagic food web requires a reliable diet matrix that accounts for these diel migrations. And we've published 3 diet papers so far. Becky's now working on a 4th paper. And to date, we've integrated stomach data from over 26,000 predator stomachs, including original stomach sampling by my lab and by Steve's lab. So Atlantis has a long runtime and has always, and this has always prevented probabilistic simulations. However, innovations in our lab utilize the diet statistical method that I described earlier. And so we ran 1,000 simulations using Microsoft Azure Cloud Computing resources, which is equivalent to 3.2 CPU years of computation time. And then the output from these 1,000 runs, which is sampling, the error range described by that statistical method in the diet matrix. We ran 1,000 of these runs, and these were used, the outputs were used to train these 6 types of statistical emulators, which basically predicts what Atlantis would say under different variations of the diet matrix. Obviously, this is a very large parameter space to sample, and so we used an efficient method called Latin hypercube sampling to sample the parameter space. And on the right here of the slide, you can see the resulting range of uncertainty in the Deepwater Horizon oil spill recovery trajectories. This was published in 2018. This is a new project that's funded by the Florida Restore Act Centers of Excellence program through FIO. This work is being done by B. Combs as part of her PhD dissertation. We're using Atlantis to examine the marginal value of seagrass habitat to turtles, birds, and mammals. You can see our focal species here. And Tuan Minh Hu is helping to provide a harmful algal bloom series, which we'll use as a pulse disturbance to analyze community resiliency. As seagrass coverage changes, you can imagine that the carrying capacity of focal species may follow a saturating curve like this one. The parameters describe the value of seagrass habitat and the decreasing marginal benefit as other factors begin to limit population growth. So Atlantis will be used to help sketch out these relationships. Kelly Vassbinder is studying larval dispersal patterns in the Gulf of Mexico. She has used CMAP ichthyoplankton sampling data, which uses a mock nest sampling net that allows you to record the depth that each particle was captured at. So the larvae are captured at some known age, which she determined based on their length by developing original larval growth models, which are now published. I'm not going to get into that here, but it was a really neat paper, as you can see evidence of different life history strategies between different families. Kelly developed her own Lagrangian transport model, which utilized hydrodynamic data provided by Bob Weisberg and Yonggang Liu. And she ran the movement of the larvae back to the origin to identify spawning sites, and she ran them forward to the end of the pelagic larval phase to identify settling sites. I don't recall how many species exactly she did this for, but something like 20 or maybe more. So the really cool thing, one of the points of innovation here was that we didn't assume surface transport as is often done. She developed generalized additive models to estimate the depth of larvae at different ages. And so she was able to use the hydrodynamics in 3D dynamically with realistic vertical migration and represents which currents the larvae were using to disperse at different stages of development. This is my most recent project. Chris Stallings and I are working together to examine the value of artificial reefs as management tools. He has a number of natural and artificial reefs near Tampa Bay that have a long time series of community abundance observations from divers and video monitoring. And my new student, Tiff Ratzel, is going to be developing a food web model of natural and artificial reef communities. We'll examine the community composition as well as resiliency of the reefs to environmental disturbance. We hypothesize that natural reefs may be more resilient if they have a more biodiverse community, particularly among small and cryptic invertebrates. Nevertheless, artificial reefs may replicate many of the ecological functions of natural reefs and serve as a biodiversity reservoir and a useful management tool. This project is using a model called Ecopath with Ecosim, which is a mass balance food web model, quite a bit simpler than Atlantis. And this provides a segue to my last slide. My lab in December of last year hosted a number of meetings, including the Ecopath 35 Symposium, which drew attendance from 110 scientists from 25 countries around the world. It included 5 days of training workshops and a keynote presentation from Bob Ulanowitz, who's a University of Maryland emeritus professor and inventor of ecological network theory. All right, that's everything. Thank you very much.
Very good. We have time for questions.
I'm not sure how to stop sharing.
I see you're working on another manuscript.
Oh yeah, of course.
Yeah, unmute yourself if you have a question. Or if not, I have a general question, Cam.
Sure.
So how easy or how difficult is it to apply the model to a new environment? If a new student wants to learn.
Well, you know, I mean, Atlantis is a lot of work. It's you know, there's a lot of you know, it takes in chemistry and physics and biology, and there's a lot of things that need to be sort of tuned, and it takes a while to use. So I typically only will subject a student to that if they're doing a PhD. I often, I mean, you know, these other statistical methods and stuff like that, but a more realistic model for a short-term project is Ecopath and Ecosim, because it's just the food web, and it's a little more, yeah, not as much can go wrong. Atlantis is yeah, you need a little bit of yeah, it can conceivably take quite a while before you get sensible results out of a model like that.
Okay, yeah, that's what I figured. So basically that means you can't take summer interns.
No, no, and plus it's also a much different ball game in terms of the you know, how user friendly the models are. I mean, Ecopath, it's got published manuals online. There's hundreds of examples. There's models you can download. It has a nice graphical user interface. I teach a course on that, by the way, in alternating years, as I'll be teaching next year. Whereas Atlantis is all operated with text files, and there's a lot of programming. Anytime you want to get data in or out of the model, you basically have to write a program that will interface with the you know, with the data files. So it's an investment.
There's a learning curve, or always. Okay, thanks. Any question? This is my last call.
Oh, I see.
Looks like
Steve has his hand up.
Yeah, thanks, Cam. Thanks for that sort of view of all things Atlantis. And I know that there are a number of these implementations around the world. Is kinds of modules are being developed for
Your internet connection is not good.
I'm sorry, Steve. You're going to have to repeat that.
Maybe you can turn off your video, Steve. Well, did I hear another question? Okay. Well, Steve is
yeah, sorry, Steve, but now your video is frozen as well. So I don't know if you can hear me, but I can't hear you.
Well, Steve, you may ask Cam offline, and let's pass this. If there's no more question, we move to Brad. Sorry.
I see a question, but
looks like Kristen has a question.
Yeah, I heard something, but
Kristen.
Who had a question? Kristen or
Kristen?
someone? Can't hear anything. Let's move forward. Thank you, Ken.
Okay, thank you.
Looking forward to you. So let's first see if Brad can share his screen. See if it works. You may request control.
I no longer see the request for control sign. Let's see if.
Cam, please turn off your screen sharing.
Yeah, I don't see the request control.
Yeah, I'm not sure how to turn this off.
How
Can
did you turn on? It's the same button. You just click again. It's the on-off button.
Okay, so is it off now?
I don't see it anymore.
Yeah. So, Brad, do you see any request control button?
I don't. I did earlier.
I don't think you need to request control. I think you can just share content. I have the ability to just go ahead and click the share content button next to the mute mic button.
It's not, it hasn't let me share content all day, so I thought if I could request control, that would maybe fix that problem.
Michael Schramm said request control only works when someone is actively sharing. So I think, Dr. Hu, you have to share your screen, and then Brad might be able to request it.
All right,
That'll
do
work.
that. And let me see, where's the PowerPoint code? Sorry for the technical problem. I'm trying to find the PowerPoint to share. Wait.
It's in the last email I sent you.
Here.
All right, now I can request control. All right.
I allowed. Now, do you have the control?
Doesn't seem like it.
Okay.
All right,
So
maybe I do. It's just slow.
Yeah, I see a moving mouse. Is that yours?
That's mine, I guess.
Okay, so you can control. Try to hit next page.
I'm doing that. It's not going anywhere. Let's see.
Okay. I'm sorry. It doesn't work. So I have to be your pointer or
okay.
page turner.
All right. Now I've lost it.
Okay, so
there.
I run the slides for you then.
Okay, thank you.
And, let me try again. All right, I'm here.
Very good. All right, so I'm just going to share a project that started during the last lab meeting we held in person before the pandemic hit, and then continued as weekly Skype calls for the last several months, and ended up with a paper submitted at the beginning of August. So it's been a fun little thing to keep us all occupied. So
sorry to interrupt.
Sure.
So Brad Sable, Dr. Brad Sable is a biological oceanographer in our college. You haven't known him yet. And this is the introduction of his recent work on hypoxia and oxygen. So
thank you.
I didn't hear you introduce yourself.
Yep, my oversight. Thank you.
All right.
So the basic question is, what is hypoxia tolerance and how do we measure it in marine animals? Generally speaking, well, there are a lot of metrics you can use to assess hypoxia tolerance. You can do lethal tolerance curves where you have to kill a whole bunch of animals to see what oxygen level 50% of them survive, or at what oxygen level do 50% lose equilibrium if you're talking about fish or other animals that have equilibrium. But these are all problematic and involve a time element as well. So probably the most widely used metric of hypoxia tolerance is called the critical oxygen partial pressure, which is putting an animal in a respirometry chamber and sealing it and measuring the oxygen over time. And you can calculate the rate of metabolism as the oxygen drops. And when the partial pressure of oxygen reaches a critically low level, that rate of metabolism will fall off. And so the peak crit is that inflection point. And that inflection point is widely considered to be a measure of hypoxia tolerance. So if you have a higher peak crit, you're less tolerant of low oxygen. So this quote from a paper by Mandic et al. Suggests that organisms that are able to maintain a routine metabolic rate at lower oxygen tensions can exploit more oxygen variable environments. And that's sort of an assumption that is not widely tested. It's just sort of assumed. So next slide, please. The critical oxygen level has been measured at least for the last 70 years or so. This is a paper by Fry, Canadian fish physiologist, where he looked at goldfish and the relation of oxygen consumption to temperature and oxygen. And he measured both the critical oxygen level for the standard metabolic rate, that lower dashed line in that figure, as well as the critical oxygen level for the maximum metabolic rate, which is measured in this case by putting a stirring mechanism in a chamber and forcing a goldfish to swim against a current. And so he referred to the critical oxygen level for the maximum metabolic rate as the incipient limiting oxygen level. And for the standard rate, it was the incipient lethal oxygen level, or the level of no excess activity. So at that lower critical oxygen level, there's no scope for increased activity any longer. So even though he measured these things, he was aware that that data doesn't necessarily refer to oxygen levels that the animals actually see in the environment. He says the worth of these data to the ecologist ultimately depends on proof that they have real significance as values limiting activity in nature. Next slide, please. We've used the critical oxygen level to assess biogeography, species biogeography in a number of cases, as well as vertical distribution in zooplankton. So it is clear from these that the critical oxygen level has some ecological component, but does it actually correspond to the lowest oxygen level that animals see in their environment, or some other metric is still a little bit unclear. Our recent work suggests that animals require at least an environmental oxygen pressure of about 2 to 3 times the critical level in order to sustain a population. Next slide, please. And we've shown over many decades, my graduate advisor in particular, Jim Childress, did a number of experiments over many years in different regions and showed that animals from oxygen minimum zones tend to have lower critical oxygen pressures than animals from shallower coastal environments, and animals from lower oxygen minimum zones, more pronounced oxygen minimum zones, such as in the California current, have lower critical oxygen levels than those from the Gulf of Mexico or from Hawaii, for example. So it's clear that the critical oxygen level is telling us something, but there's clearly still a lot of variation in both of those groups, the coastal species and within the oxygen minimum zone group. There's a lot of variation that is not easily tied to environmental oxygen level. And just to point out that that y-axis is a log scale, so that variation is substantial. Next slide, please. That doesn't stop people from making post-hoc explanations. If you find an animal that has a lower critical oxygen level than some other animal, then you will claim that it is hypoxia tolerant and it must be encountering oxygen somewhere in its environment. One of the most notable examples is nautilus. This is an animal that lives in the tropics around the Philippines and some other places where it doesn't actually see low oxygen as far as anybody's measured. So this inset figure on the left shows the distribution of the animal. Those blue points are where nautilus has been observed. This is from Obis. And the distribution of the animal doesn't come anywhere near the critical oxygen level, which is that dashed line at the bottom. It does correspond to that 2 to 3 times level that I mentioned earlier, that the environmental oxygen in order to sustain a population is typically 2 to 3 times that critical oxygen level. So for coral reef species, similarly, they measured low hypoxia tolerant or low critical oxygen levels and suggested that it reflects nocturnal hypoxia that they must see in tide pools or in little microhabitats inside coral branching. So there's a lot of post-hoc explanations that are not well supported. Next slide, please. There was a paper that came out just a year or so ago by Chris Wood that attempted to completely get rid of the critical oxygen level. He thought it was a complete fallacy and had a number of criticisms. One is that it depends very much on the way it's determined. So these graphs show the same species measured using 2 different respirometry methods. One is the closed chamber method that I mentioned earlier, where you seal an animal in a chamber and measure oxygen over time. The other is intermittent flow, where you flush the chamber periodically to maintain the CO2 level and to eliminate other metabolic waste products. And it's thought to be less stressful, and so they recorded using these 2 different methods and found that the critical oxygen level was different between them. And you'll notice that the way they determine the critical oxygen level in this case is called a broken stick method, where you just create 2 regressions through the partial pressures above peak crit and the partial pressures below. And so it's somewhat subjective, but you can see that if there's a slope to that relationship at higher oxygen values, that you can get very different peak crit estimates. So they measured two different, very different peak crits using the same species and just these two different methods. Next slide, please. There's a lot of different ways to analyze any given run as well. This broken stick method that I mentioned. Some authors apply nonlinear statistics. They'll calculate what is effectively a KM, like in Michaelis-Menten enzyme kinetics, and it's not clear exactly what that would tell you, but it clearly doesn't fit this particular data set very well. Others determine a standard metabolic rate at high oxygen levels and then extrapolate that, and where it intersects with the lower oxygen dependent part of the curve, that's the critical oxygen level. So you can see with these different analytical methods, you can get a peak crit that varies from 2.8 up to about 9 almost. So a lot of variation. All right, next slide, please. The other criticism that Chris Wood levied against peak crit is that there's no theoretical justification for the concept that this 1 point, the critical oxygen pressure, carries minimal information content. And he actually lobbied for measuring something that characterizes the entire curve from high oxygen to low oxygen rather than distilling it down to a single point. It's not clear to him why that 1 point should have physiological significance. So this graph shows, different labs measuring the same species, and they come up with very different curves and very different critical oxygen levels. So there's clearly, this is Fundulus, which lives in a lot of different oxygen environments, that it would explain some of it, but there's a lot of different methods between labs that explains a lot of that variation. Next slide, please. So this was our project. It includes, as authors, all of my current lab members, who I'll introduce in a moment, as well as a former lab member, Matt Burke, who you may remember graduated here a few years ago. And we submitted this on August 3rd to Functional Ecology. Next slide, please. Oh, I guess I eliminated the introductory slide. Since I sent this to Chuan Min over email, it was large with the pictures, so I apologize. I eliminated my lab members. Sorry about that. But they have all contributed data to this project. Alyssa Andres is working on sharks. She's measured peak crits in dogfish shark, juvenile bull sharks, and blacktip sharks. Alexandra Burns is measuring shrimp. We have Matt Burke, who contributed some squid data that he measured while he was here at USF. Tracy Shaw has contributed krill data, as well as her special editing skills. My two new students, Christina Welsh is working on larval fish and has a manuscript in preparation specifically on larval fish already that she's been working on over the last several months. And Alexander Timpe has aspirations to work on lionfish, but he has analyzed a lot of different data sets for this paper. So, okay, so this slide shows in blue, the standard metabolic rate. These are just schematics. So standard metabolic rate declining, maintained at a stable level over time as the oxygen in the chamber declines down to a critical oxygen level. And on the right is the standard metabolic rate at any point divided by the partial pressure at that point. So obviously, if the metabolic rate is the same and oxygen is declining, then that ratio is going to increase. And that ratio is effectively the oxygen supply, the amount of oxygen supplied per unit time per available unit of partial pressure. If you increase the metabolic rate, next slide. So this is more of a routine metabolic rate instead of standard. Standard is a resting fasted metabolic rate, and this is routine, where we don't control activity levels. The oxygen supply starts at a higher level, but increases up to that same point. And then next slide. Maximum metabolic rate is measured in swim tunnels or after an exhaustive chase in some cases. And again, it starts at a higher level and increases as the partial pressure drops in the chamber. and all three cases reach a maximum oxygen supply that we're calling the oxygen supply capacity, alpha, which is characteristic of that species under those conditions. And that oxygen supply capacity describes the slope of that dashed line on the left, which is effectively the critical oxygen pressure for any metabolic rate. So you can calculate what the peak crit is for any rate if you have measured the maximum oxygen supply capacity. And in discussing this relationship, it occurred to us that rather than trying to guesstimate from the various methods where peak crit actually is, we can calculate alpha, the oxygen supply capacity, during those runs and see where the oxygen supply capacity actually reaches its maximum. Next slide, please. So these are inverse or reversed from the previous slide. So on the left is that oxygen supply increasing. So the metabolic rate divided by the PO2 at that particular point increases up to a maximum and then declines. In some cases, it plateaus. But you can see that the maximum there, 0.027 micromoles of oxygen per gram per hour per kilopascal, is the highest that it achieved. If you then divide the metabolic rate, so each of those blue points on the left, if you divide that metabolic rate by alpha, you get that straight red line. So that's the peak crit for any metabolic rate. And you can extrapolate that up to the maximum metabolic rate. For example, if you assume that the peak crit for maximum metabolic rate is air saturation, you can calculate what that maximum metabolic rate would be. And the dashed lines there show the most widely accepted method for determining peak crit, where you calculate a standard metabolic rate and extrapolate it to the oxygen-dependent part of the curve. And so the peak crit calculated that method, and this, what we're calling peak crit alpha, correspond very closely in this case. Next slide, please. So these two curves that I showed earlier, where they compared closed versus intermittent flow respirometry and came up with two different very different peak crit values. If you plot them using this new method, you can see that the metabolic rate is higher in the closed system measurement, possibly due to increased stress or something of that nature, but the oxygen supply capacity alpha is the same for that species using those two different methods. Next slide, please. And again, this should predict maximum metabolic rate. So on the right are two different individuals of the same species. The one is maintained at a standard metabolic rate and reaches a peak crit at that red line. And the other was exercised, and you can see that the maximum metabolic rate falls along that line as the oxygen partial pressure declines. So this is the schematic on the left is something that I published earlier this year, and it is basically a previously unrecognized relationship between these various parameters. The maximum metabolic rate, basal or resting standard metabolic rate is BMR here. The peak crit for that maximum metabolic rate and the peak crit for that basal or standard metabolic rate are related mathematically, and so you can calculate one from the other three. And if you do that calculation for most of the species that have been measured or for which those data exist, peak crit max is very close to air saturation for most of those species. Next slide, please. So using our new peak crit alpha method, we compared all of the data that we could find in the literature. Most of the reports will give a peak crit in the text or in a table, and then they'll show what they call a representative curve, a representative respirometry curve, which is probably usually the best looking curve of all of them that they measured. And so this may not be the most fair representation, but Using that data that we were able to extract from those representative curves, the Perit alpha method that we developed correlates very strongly with the Perit that they reported using other methods. And the various methods that other people have used correlate more or less strongly. The one using the standard metabolic rate extrapolated over to the oxygen-dependent part of the curve correlates best with our Perit alpha method. So previously, Perit was defined as the oxygen pressure at which standard metabolic rate becomes oxygen-dependent, and we've redefined it as the Perit at which the PO2 at which oxygen supply capacity is maximized. And this provides a much more physiologically justified definition that clarifies exactly what Perit is telling us. It's not a measure of the hypoxia tolerance of the animal, it's a measure of the maximum oxygen supply, which doesn't evolve in most cases for low oxygen, it evolves to support maximum metabolic rate. Next slide, please. One little hiccup we noticed is that for roughly 5 to 10% of the curves we were able to find in the literature, the oxygen supply would increase continuously up to the lowest oxygen pressures measured. And in some cases that would have a, I'm pointing to my screen, you would have a positive y-intercept, like in that top left figure, which indicates that there's oxygen being consumed even when there is no oxygen left in the chamber, which is obviously not possible. So this indicates error in the PO2 measurement, most likely due to calibration error. It's very difficult to calibrate a true zero, and in most cases it doesn't matter too much because most species that are measured don't get to oxygen levels quite that low. But if you're measuring things from oxygen minimum zones, it can become a real problem. And because the oxygen supply capacity is a ratio, it tends to escalate very rapidly as you approach zero oxygen if there's any error in that measurement. So Alec Timpe, one of my new graduate students, developed a very clever method that would require an additional 30-minute talk to explain, but it effectively corrects for that calibration error and greatly reduces the oxygen supply capacity estimates in some cases. All right, next slide, please. So that's going to be a separate paper that Alec is preparing now. So this is just an example to show that using this new method, it doesn't require that you have a constant metabolic rate. Previous methods were much more reliable if the animal maintained a very steady metabolic rate at high oxygen pressures. But in the present method, that doesn't matter. So this is a larval anglerfish, just one individual that I caught off Hawaii, November 2018, and measured its respiration rate, and the animal swam continuously, seemingly continuously, which surprised me. I would have expected them, like the adults, to largely sit in place and do nothing, but it swam very actively, which led to these fluctuations in the metabolic rate over time. But even though the metabolic rate was not constant, it did reach a maximum oxygen supply at low PO2, which allowed us to calculate a critical oxygen level for any metabolic rate. So if you count the lowest rate achieved during that run as the standard metabolic rate, you can calculate a Perit for the standard. And the highest, we'll call an active metabolic rate, has a Perit near 4 kilopascals, which is very close to the lowest oxygen level that this animal would see in nature. All right, next slide, please. So Perit is telling us something about low oxygen exposure, but from Perit alone, you cannot tell if that is an adaptation for hypoxia tolerance or for athleticism. So these are two hypothetical species, 1 and 2, in blue and black, that have the same standard metabolic rate, but Species 2 has a much lower Perit, which means it has a higher oxygen supply capacity. So it may be that that species is exposed to persistent hypoxia like an oxygen minimum zone species would be, and it has a lower Perit for maximum metabolic rate as well. But it may also mean that it never sees hypoxia, but it's just a more athletic species. And so it could have a much higher maximum metabolic rate that is supported by that oxygen supply capacity. So without measuring the maximum metabolic rate, it is impossible to say with certainty whether the Perit or oxygen supply is an adaptation for low oxygen or not. All right, next slide, please. So species that are exposed to persistent hypoxia tend to have a higher oxygen supply capacity at a given metabolic rate. So the oxygen supply capacity is strongly correlated with the maximum metabolic rate achieved by a given species. So each of these points on this graph are different species, mostly taken from the literature. So there's strong correlation between metabolic rate and oxygen supply, as you would expect, and then hypoxic exposure elevates the oxygen supply capacity by some fraction that depends on the oxygen that they're exposed to. Next slide, please. All right, so in the last few minutes, I wanted to introduce a new paper that's based on some of these same ideas, and this is very much in preparation. Some of this data I just put together this week. So there's a common perception that oxygen supply is limited by high temperature, and that results in reduced aerobic scope at high temperature. So this graph shows the maximum metabolic rate in black and the standard metabolic rate in red. Standard metabolic rate tends to increase with temperature faster than maximum metabolic rate. So at some high temperature, those two would meet. But normally that's well outside the range of temperatures that animals actually live at. But they do reach a point where the maximum metabolic rate is roughly 2 to 3 times the standard metabolic rate, which seems to, again, be a population limit. So whether or not this decline in aerobic scope is related to oxygen supply is a very commonly pushed theory, but is really not supported by very much data. So next slide, please. So this is another schematic showing, again, two different, well, it could be the same species measured at 2 different temperatures. So the maximum metabolic rate increases with temperature, but at that higher temperature, coastal species would still be exposed to air saturation, 21 kilopascals. So the Perit for maximum metabolic rate is the same. Because standard metabolic rate tends to increase a little bit faster than maximum metabolic rate, the Perit increases. But it's really the oxygen supply capacity that changes with temperature to meet the maximum metabolic rate. So at high temperatures, maximum metabolic rate increases and oxygen supply capacity increases to meet it. And so the idea that it's limited at high temperature doesn't fit with this idea. Next slide, please. So this is a brand new sort of conglomeration of data. These are the temperature sensitivities for standard metabolic rate along the x-axis and for Perit along the y-axis. And normally, as I mentioned, standard metabolic rate has a higher temperature sensitivity than Perit. But for all of those blue dots, which are vertical migrators, diel vertical migrators, similar to the ones Cam was referring to in his talk, myctophids and krill, for those, the temperature sensitivities for Perit and standard metabolic rate are the same. So that gray dashed line is the 1-to-1 line. They fall directly on that line. And what that means is, if you look at the equation in the bottom right, because oxygen supply capacity is equal to the standard metabolic rate divided by Perit and equal to the maximum metabolic rate divided by its Perit, then If the temperature sensitivities for SMR and Perit are the same, then the temperature sensitivities for MMR and its Perit must be the same, which creates some interesting interactions. Next slide, please. The oxygen supply capacity is higher in the species from the eastern tropical Pacific, where the oxygen is much lower, than it is for vertical migrators in the Gulf of Mexico or Hawaii. I think the California current system, they're slightly different species and not strongly represented by the same sort of vertical migrators. Otherwise, I think they would also fall above the others. So they do have a higher oxygen supply capacity, but they all have similar temperature sensitivities. Next slide, please. So what that means on the right is similar to the schematic I showed earlier. This is actual data from black sea bass. So maximum metabolic rate increases with temperature from 12 to 27. The Perit for maximum metabolic rate stays near 21 kilopascals air saturation. And as a result, the oxygen supply capacity increases to meet that maximum metabolic rate. If oxygen supply capacity doesn't change with temperature, like the vertical migrators, you get something like you see on the left, where Perit and maximum metabolic rate change with oxygen partial pressure. They go up and down together across that temperature range. So what that means is as temperature drops, the maximum metabolic rate drops by some amount, and the Perit for maximum metabolic rate also drops. Next slide, please. So this is a little bit complicated, but I'll try to explain it. So these are 3 different regions with slightly different oxygen characteristics. The blue curve shows the oxygen partial pressure declining with depth. In the eastern tropical Pacific, it's a very pronounced decline. In the Gulf of California, slightly less so. And on the right, the California current has much higher oxygen levels, but they still decline to fairly low levels by the time you get to 500 meters. We don't know what the temperature sensitivity for maximum metabolic rate or Perit max is, so we can model that. So the numbers next to the black lines are the temperature sensitivities, 0.3, 0.8, and 1.7. 0.3 is pretty typical for most animals, so these other 2 are much higher temperature sensitivities. So if the Perit max declines with a low temperature sensitivity, then it's going to be oxygen limited because the oxygen partial pressure is always lower than Perit max. In the Eastern Pacific, that's also true even if the temperature sensitivity is very high. So no matter what, the Perit max and oxygen and the maximum metabolic rate are oxygen limited, and they will decline with depth exactly as oxygen does. In the Gulf of California, same sort of idea, but animals are going to be oxygen limited at the 2 lowest temperature sensitivities, but temperature limited if the temperature sensitivity was very high. So that means that the maximum metabolic rate would still be very low at depth, but it wouldn't be oxygen limited. And in the California current, no matter what the temperature sensitivity is, in that upper 2 or 300 meters, animals would be temperature limited, no matter what the oxygen does. All right, next slide, please. So the only data we have are for the jumbo squid, which does vertically migrate, and its temperature sensitivity is pretty close to that 0.8, which is the median one that we estimated there. That's that top red line. Next slide, please. So this shows the estimated factorial aerobic scope on the right. So anywhere where factorial aerobic scope is greater than 2, we considered metabolically available habitat. And in the top panel is a low temperature sensitivity down to the bottom panel, which is a much higher temperature sensitivity. And you can see that that orange, which is where animals could theoretically live, is, so this is across latitude from 60 north to 60 south, and as a function of depth in the upper 500 meters. So with a low temperature sensitivity, the jumbo squid could live anywhere pretty much except at depth in the eastern tropical Pacific. It becomes oxygen limited. But as the temperature sensitivity increases, it is limited to the upper few hundred meters of its native tropical habitat. So that middle panel showing 0.8, that's the temperature sensitivity that we've actually measured for Dispersal, which suggests that it can only live where it does currently. It would be temperature limited in cold water. So as you move toward the California current, its aerobic scope would drop below 2 and it would not be able to sustain a population there. And this is consistent with observations that Dispersal expands its range into the California current following warm water events such as El Niños. All right, I think I will stop there. Thank you.
Thanks, Brad. Let's see, we have a few minutes before we go to break for questions.
I realize that can all be very confusing if you don't spend your waking hours thinking about it. Question by Catherine.
Can you hear me? Sorry.
Yes, I can hear you now.
I guess I'm wondering if there is a way, or if there have been efforts to study on a population level the Perit, and because it sounds like all the study or the methods involve working with individuals in labs and then like 2 times 2 to 3 to estimate the Perit for a population level. So I'm wondering if there are efforts to do that, or what methods would be involved.
So the way we've done that is by mapping what we either call the metabolic index, or we've shown more recently that it is equivalent to the factorial aerobic scope. So if we know from individual animal laboratory measurements what the temperature sensitivities of those are, we can say what the factorial aerobic scope would be at any given parcel of water. And then we compare that to where the animals actually live and where they stop living, where you don't find them any longer. And in most cases, once you get to a factorial aerobic scope of 2 or 3, the population ceases. I don't know how else you would measure that at a population level. Yeah, that's
okay.
It's a good question. But
no, I'm just like wondering how much of an assumption that is. I don't know this discipline well enough to make that judgment, but yeah. Well,
there's certainly error in any of these measurements, and the more species and the more individuals of each species, the more temperatures you have, the better you can refine it. And so probably the best example that we have that it does actually contribute to distribution changes, or for these zooplankton in the eastern tropical Pacific, there are species that live at this lower oxycline where the oxygen minimum zone just starts to creep up again, and with very small changes in oxygen of about 4 micromoles, we saw differences in the abundance and distribution of species from that, which correlated very strongly with the critical oxygen levels that we had measured.
Nice.
So.
Okay, thank you.
Yep, thank you.
Maya, I think you're next.
Hey, Brad. I've heard a couple of things kind of up and down over the past few years about this, so I was wondering if you could comment. But I've kind of heard this notion that gelatinous organisms are gonna do better in a lower oxygen world. Is there any consensus on this, or who do you think might be the winners in the future game?
That's a very good question, and I can say that there are very, very few data for any animals that don't have complex circulatory systems. For whatever, for I guess obvious reasons, physiologists have tended to focus on animals that have hearts and lungs and gills and things like that. So we do have enough data to know that the metabolic rate is regulated to some extent in jellyfish and ctenophores and things like that, meaning that they can maintain a constant rate down to a critical low oxygen level, even though they don't have an obvious way to transport oxygen to their tissues. So we assume that it relies on diffusion, and their rates are just so low that they're met until some critical oxygen level. They may have behavioral ways to regulate. But as far as whether or not they will be winners and losers, I don't think there's any real evidence for or against that. Most of what I've heard, like I can say that the idea that they'll be winners in ocean acidification studies, there was a study that tried to link their distributions to CO2, and that was debunked. Jellyfish blooms have largely been debunked as an artifact of increased sampling. So there's no real evidence, as far as I know, that their populations are increasing or doing better already, and no reason to think that they would do better. So.
Well, thanks.
Yep.
Well, any more questions? If not, we're going to have a short break. And thanks, Brad.
All right. Thank you, everybody.
Yes. So let's come back at 3:10. Then we are right on schedule. Thank you. Enjoy your coffee. So, Brad.
Yes.
Okay. So while others are enjoying coffee, here is my additional question, following Maya's question. So I heard there's a particular zooplankton in the Great Lakes called Holopedium. That is a low oxygen tolerant species, which outcompetes the other zooplankton. So people have seen rising of a holopedium in Great Lakes, but that's not the ocean.
Yeah.
But in the ocean, there's no similar increasing of whatever species that is tolerant to acidification or decline in oxygen.
I mean, there may very well be, and we just haven't figured out how to detect it yet. I think because the ocean is so much larger. But do they know why that particular species is out competing? Is it because they think of the oxygen tolerance?
Because of the increased acidification.
Oh.
So this species doesn't require calcium, or requires a very low calcium level to build their shells. So it outcompetes other zooplankton. I thought that's very interesting.
Yeah, well, a lot of people have speculated that the jumbo squid invaded northern waters because it either got too hot for them down south, or they were taking advantage of other species that had been overfished in the California current waters. There's been at least as many different hypotheses as there are researchers working on squid. And I think that it's entirely due to a temperature limitation that's relieved. But it's very complicated because they, I think their reproductive mode comes into play, that they may not be able to establish a reproductive population in northern waters, even if they weren't temperature limited there for various reasons. And so I don't know, it's always complicated, I guess, is the take home.
We just need more data.
Yeah, I mean, that's the limitation in this approach, is that we can say where animals can live, we can't say where they do live. So it's a tricky thing.
All right, thanks.
Thanks for your question. I'm gonna go grab a coffee.
Yes, please.
Chuanling, can I try sharing just to make sure?
Sure, go ahead. Yeah, so put your first slide on screen, and good, very good.
Okay, perfect.
All right, so we wait until 3:10.
Maya, can I also give it a go quickly? Just, I don't want to kick you off, but can I just check I'm working all right this end?
I think you should be able to do it now.
Thanks, Maya. It'd be just my luck that mine doesn't work right.
That looks good.
Is that looking okay your end? Awesome.
Yep.
Okay, I'll give it back to you.
Oops.
Okay, I hope everyone is back, and let's get started on the last session of today. So we're going to have two speakers talking about viruses, tides, glaciers, all interesting topics. The first one is given by Dr. Maya Breitbart on her virus studies. So Maya, please take it away.
Yeah, thanks, Tuan-Min, and thanks to everybody for hanging in there. I know it's been a long day. I'm going to try to shake this up a little bit at the risk of technological disasters, but you'll see in the chat window that I pasted a link to a poll, which I believe is active. Someone please speak up if it's not. And I want you to just enter into this poll one word, or if you're doing multiple words, put a dash between them so they stay together, about what comes into mind when you hear the word virus. Well, I have to say I'm pleasantly surprised that it wasn't just dominated by coronavirus on the whole thing. But you can see there are a whole lot of negative connotations here. And this is truly how most people think about viruses, as small things that make you sick. They are certainly ruining our lives right now. But these are not the ones that I'm going to talk about today, because obviously I'm going to focus on the ocean. But there's just a couple questions at the beginning of the talk. So choose a choice, A through D, for what you think the average diameter of a marine virus is. You know they're small, but how small are they? Okay, good. So the consensus is definitely on the small side, and you guys look about split. So all the answers to these questions will be in my talk. I'm not going to spoil them now, so I'll keep you in anticipation. Look about 50-50 picometers, nanometers. There are a lot of viruses in seawater. How many do you think there are in an average milliliter? Okay, again, pretty equally split. Looks like 10 billion is winning right now, which is not the correct answer, but you'll see that in a minute. Just a true or false for you. Are there more, true or false, there are more viruses in the oceans than grains of sand on Earth beaches? True is definitely winning this one. And then I will leave you with the most controversial question in all of virology. Are viruses alive? Okay, also a very even split there. Okay, so that is it for the polling. You can go back to your team's window, and I'll kind of jump into my talk now. So I'm going to answer actually all of those questions for you in this talk, with the exception of the last one, whether viruses are live, but hopefully I'll give you enough information for you to make that judgment on your own. Technically, it really comes down to what you consider a definition of life and the stage that the virus is at. So today I want to talk to you about marine viruses, and I'm just going to kind of start by zooming out, because you've heard a couple of talks today about other biology in the biology in the oceans across all size scales. And what we know is that biology in the oceans spans 9 orders of magnitude in terms of size. So on kind of the upper size scale, where people are most familiar, you have things like great blue whales, which can be 30 or so meters large. And most people, especially snorkelers and divers and marine enthusiasts like you guys, know that are very familiar with kind of these things on the bigger end of the spectrum, right? So whether it's fish, or they're copepods that eat them, etc. But what my lab really focuses on is from the bacteria and kind of downward for the most part. Occasionally we'll study giant things like fish eggs, but everything today is going to focus on the microbes. And one important thing that I want to point out is that oceanographers really heavily rely on a differentiation between particulate organic matter and dissolved organic matter. And typically this size cutoff is defined at 0.2 microns. So anything smaller than that is called dissolved organic matter, and above that is called particulate organic matter. And hopefully you're starting to see where my dilemma and my title of deciphering the dissolved comes in, because the things that I study, viruses, the majority of which are phage, which are viruses that infect bacteria, are over here in this dissolved size. And while they are technically that small, they are definitely not dissolved. So just to kind of bring you back to the abundance, viruses are the most abundant biological entities in the oceans. So what I've shown here is a sample of seawater that we pulled down onto a 20 nanometer filter. So we're capturing the viruses, the bacteria, and everything larger. We've then added a dye that binds to all DNA and RNA, which actually lets us see things that are smaller than the resolution of light microscopy. And then we look at that under fluorescence microscopy. And what you're seeing in this image here, these bright larger dots are bacteria, and these small pinpricks of light are viruses. And then on average in seawater, surface seawater, we have about 1 million bacteria per mil and 10 million viruses per mil. Since bacteria are the most abundant hosts by far in these environments, the majority of the viruses are phage, so viruses that infect bacteria. And just to give you a fun comparison, in case you've got your kids at home learning along, or you want some dinner conversation tonight, there are only about 10 to the minus 19th great white sharks per mil of seawater. There are by far a lot more microbes than anything else. In just 1 liter of surface seawater, there are more viruses than there are humans on the planet. So if you took all of humanity and drowned them, or more nicely, took them swimming in the oceans, they wouldn't even be on the same scale as the number of viruses. In addition, in the oceans, there are 100 billion times as many viruses as there are grains of sand on Earth. So anybody who put there were more viruses was correct on that one. Even though these viruses are extremely small, with an average diameter of about 50 nanometers, kind of head to head on their sides, if you lined all of these viruses up, all the viruses that are in the ocean, and you line them up, they would extend for 12 trillion round trips between the Earth and Mars. So just a huge, huge number of viruses to be thinking about. So why should you care? And what are all these viruses doing out here? And so here's an image that Natalie Sawaya in my lab prepared for a paper we published in Nature Microbiology, kind of going through the roles of viruses in the ocean. And so I'm going to walk you through this. Just on the right-hand side is the traditional food web. So this is typically what everybody is brought up thinking about, where you have large eukaryotic phytoplankton that are doing most of the primary production. Those are eaten by grazers, which are eaten by larger things, which are eaten by larger things, and on it goes. But what we actually know is that the microbial loop, the other side of this diagram, is also very important. And about half of the photosynthesis that happens in the ocean, so half of the carbon that is fixed and half of the oxygen in every breath you take, is fixed by cyanobacteria, or bacterial autotrophs within the oceans. And so that's a major source of carbon fixation. And those can be eaten by grazers, which could go on up into the larger food web, or they can be lysed by viruses. And about 50% of the mortality of these guys is due to viral infection. And a major difference from that is that instead of kind of feeding into the larger food web, this releases a lot more dissolved organic matter. So a virus will infect these bacteria, but whether it's the photosynthesizers or the heterotrophs, they can infect both of them. And then they will inject their DNA, make lots of copies of themselves, blow up that host, releasing lots more dissolved organic matter, which can further fuel this microbial loop. So you can generally think about this as sort of the ocean's recycling system. In general, it's detracting from the biological pump and keeping carbon and nutrients in rapid cycles within the ocean. Now, there are some other ideas floating around that are receiving more and more attention right now. For example, it seems that a lot of the organic matter released through virolysis is sticky. And so that may actually accelerate the biological pump. And we're trying to figure out kind of the link versus sync rules of these cycles. So as I mentioned, phage, the viruses that infect bacteria, are efficient killers of marine bacteria. They tend to be fairly host specific, so they can structure the diversity of bacterial communities. And overall, they release about 10 billion tons of carbon per day just in the oceans. But phage are not just brutal killers. And I, in fact, like to think of them much more as the puppet masters of the marine microbial realm. And there are a few different ways that they can control or influence their host behavior. The first is that phage infection can affect the host metabolism through either expression of the phage-encoded auxiliary metabolic genes or through regulating the expression of the bacterial host genes. And so you can think of them kind of as small wizards in this way. And in fact, people have even suggested that when you're considering modeling or other analyses of bacterial communities, that you should consider uninfected cells as different than infected cells, which we call virocells, because that's how different their metabolism may be. Secondly, think of phage as little drug dealers of the ocean when it comes to gene transfer. And so phage are responsible for a lot of gene transfer through a process called transduction, which can affect both the host diversity and the function by giving them new capabilities. So all of these major rules that I've told you about so far are kind of invoked through the phage infecting the bacterial cell. And even though I'm going to kind of focus on phage today, there are viruses that infect every single organism on Earth, whether it's bugs or plants or animals, absolutely everything. But since phage are the most important, I'm going to focus on those. So what about when they're not infecting cells? When you just have a phage particle out there in the ocean, what does it mean for ecology? And so for this, I want to think about the viral particle itself, can see, I can't see myself, but I hope you can see that in the background, there's a phage that Mackenzie Kerr drew for me. And I even brought a little prop that I'm hoping you can see here. Most of my costumes and props were left at work, sadly. But viruses are these kind of very simple particles, even though they can wreak a lot of havoc and do a lot of interesting things. So essentially what you have inside is DNA or RNA, so nucleic acid, and then that's surrounded by a protein coat, as well as other structural elements that are all proteins. And so the one here is obviously just a piece of art, but the one that you see on the screen, I would argue, is an even more beautiful piece of art because that's actually what it looks like. So this is phage T4, and Tony Greco here within our college took this electron micrograph. He's just added fake colors for you to be able to see it. But you can see here, this is the phage capsid on top, as well as its tail and tail fibers that are kind of in that pinkish color. And here it's infecting a bacterial cell, which is shown in orange. Viruses are highly enriched in phosphorus and nitrogen compared to cellular life. So I'm sure in your core courses, you've heard about the Redfield ratio. And what you need to know is kind of compared to cells, viruses have a lot more phosphorus and nitrogen. So their C to N to P ratio is about 20 to 7 to 1. And other studies out of Joshua Weitz's group at Georgia Tech have shown that virus particles can account for up to 24 nanomolar of the dissolved organic phosphorus in the oceans, which could be about 5% of the DOP pool, depending on where you are. And so this is kind of where our lab started to think about what might be other impacts of these viruses. And I'm going to highlight for a second Kristen Buck because she hasn't given a talk here today, but my research drastically changed when she moved to USF because she came in, and even though Bob Byrne had been telling me this for probably decades, I think it didn't click until Kristen started and tried to convince me that trace metals are really important. If you know Kristen, she talks about trace metals a lot. And as Tim told you this morning, there are low concentrations of iron in the ocean, and it's really important to consider how available that iron is. And so when Kristen first gave her talk and talked about how more than 99.9% of dissolved iron is organically complex, the phage that is constantly living on my shoulder is kind of whispering in my ear and saying, hey, wait a minute, we're within the dissolved fraction. Could there be any interactions between iron and phage? And so I'm not going to dwell too much on iron limitation because you heard this from Tim this morning, but I just want to reiterate that iron is limiting for primary production in a great deal of the surface ocean. So on this graph, all the red dots indicate iron limitation, and you can see areas that have lower than expected chlorophyll based on the amount of nitrogen. That generally indicates iron limitation to us. And to go back to that definition of particulate versus dissolved, again, we use these operationally defined size fractions to describe iron. So if you have the total iron in the sample and you were to filter it onto a 0.2 micron filter, anything larger than that would be captured on the filter. That's the particulate iron. What goes through that filter is the dissolved iron. You can then further fractionate that by filtering through a 0.02 micron filter, and everything that goes through that is your soluble iron, while this fraction between 0.02 and 0.22 microns is the colloidal fraction, and that's really what I'm going to focus on today, because as you start to look at where biology falls into these different size fractions, bacteria typically are larger than 0.2 microns, so they would be in that particulate iron phase, and some studies have looked at potential contributions of bacteria to iron concentrations. Phage, which are of course my favorite, obviously, fall here within the colloidal size fraction. So again, that about 50 nanometer diameter puts them between 20 and 200 nanometers. And siderophores, which are well-known iron-binding ligands that are extremely strong and get a lot of research, are within the soluble size fraction, less than 20 nanometers. And so just kind of looking at all of the different things that can bind to iron and regulate its bioavailability, our phage fall right here within this colloidal size fraction. There are many other things such as the siderophores, various nanoparticles, humic substances, and many, many unknown ligands that are out there as well. But here we wanted to look at the role of phage as potential ligands. So is there any evidence for a phage interaction with iron? And a deep dive into the older literature showed us that yes, there is. And a lot of that information comes from E. coli, which is essentially the lab rat of microbiology. Using that information from E. coli and combining it with our knowledge of the oceans, Kristen and I and a master's student, Chelsea Bonine, developed the Trojan horse hypothesis, which I'm going to walk you through. So first, to start with the evidence. This is a schematic of phage T4, and structural studies have shown that phage T4 has 7 iron ions in the receptor binding tip of its long tail fiber. So if here's your phage, these are your tail fibers here. And at the very end of that is where it interacts with specific receptors on the host cell. So if you zoom in to this tail fiber protein, and then there are many copies of it, but if you look in just one monomer, you can see these 7 iron ions. And these are coordinated based on specific amino acid residues, which I'll show you on the next slide. So this is just a piece of the sequence of the tail fiber, which is called gene product 37. Don't worry about that. But it has 7 H paired histidine motifs. So this is an H, which is the histidine, then something else, then another histidine. And these paired histidines coordinate the iron atoms in E. coli phage T4, and this has been shown. Now, each phage T4 particle has 6 tail fibers, and so we would expect that that would be 42 iron atoms. So if you start doing some back of the envelope calculations on this, you come up that a significant amount of dissolved iron could be complex within phage if all phage were like T4 in the oceans. So there's some assumptions, some big assumptions that go in here. But with 6 tail fibers for phage, 7 iron ions per tail fiber, and this average concentration about 10 million viruses per milliliter of seawater, we calculate that about 10 to the 8th iron ions could be bound to phage tail fibers per mil. And that's assuming that these 7 iron ions are the only ones in the structure, which is probably an underestimate. Overall, if you divide that by average concentrations of colloidal dissolved iron, we predict that up to about 70% of the colloidal dissolved iron could potentially be bound to phage tail fibers. And so even though we need a lot more work to look at this, this shows that it could potentially be a major influence. We then went looking in the sequence databases to see if we could find these similar HXH motifs in any marine phage, because it's great to know that it's found in E. coli, but there's not a lot of E. coli phage floating around in the ocean. But what you can see here is that there are many other marine phage that have paired histidine motifs. They don't always have 7 of them, although sometimes they do. Some of them have 6, 4, or 11. And our hypothesis right now is that this number correlates to the number of iron atoms within the tail fiber. There's been some recent support that's come out of the Terra Oceans expedition that has confirmed what we thought might be happening. And so they were able to find these paired histidine motifs here in A and B in high percentages of the sequences that they got from the Terra Oceans expedition, as well as some other iron binding motifs within tail proteins that they see as conserved. And so all of this, I think, is pointing us that we are in a good direction. So here's the Trojan horse hypothesis kind of in its entirety. The idea that I've already explored with you is that phage have iron within their structures, and that could be an important ligand within the ocean. The part I haven't talked about yet is that some E. coli phage are known to use receptors that are made for siderophore-bound iron uptake as their entryway into the host cell. And this is where the Trojan horse idea comes in, because we think that phage could be using iron within their structures as a way of sort of accessing these host receptors, which are under a lot of pressure to not change, right? Because they need to be able to take up siderophore-bound iron and access enough iron to thrive. But that this kind of gives them a way to sneak into the cells. Phage DNA then takes over the host cell, and typically the phage will replicate, will kind of build all of the proteins within the bacterial cell, and then reassemble these particles, and then it will lyse the cell to release the new phage. So we hypothesize, going back a second, that the iron within the phage tails originates from within the bacterial cells, because that's where these proteins are being synthesized and assembled. And if this is true, then when the bacterial cells are lysed, instead of all of that dissolved organic matter being available for uptake, like we typically propose, if that iron from the host cell is incorporated in the phage particles, then it greatly would reduce the amount of iron available for remineralization by about 14%. So lots of ideas here, right? But how does it work in real life? And so here is work that was done by Chelsea Bonane and Salvo Caprara in Chris and Buck's lab, as well as lots of imaging help from Tony Greco. And we initially focused development of these ideas on E. coli phage, because even though we want to take this to the oceans, there are not a lot of marine systems that are nearly as well known as E. coli. And we used two phage for these experiments, phage T4, which I've shown you several times. This is the one that we have structural evidence that it definitely has iron within its structures. We also used phage T5. So we don't know if phage T5 has iron within it, but we do know that this is one that uses the siderophore receptor as its way of entering into the bacterial host cell. And our idea is that we're going to use iron 57 to trace iron from the bacterial host into the phage. So when we grow up the bacteria, we're going to grow them on iron 57 so that they're kind of saturated with this, and that's the only iron available to them. We're going to wash away all the excess iron in the system, and then we're going to look to see if that labeled iron is incorporated into the virus particles, and if we can track it into those particles after they lyse the cell. So how do we do this? Like I said, we grow the bacteria on the iron label, and then we take it through a bunch of steps after lysis to see where that labeled iron ends up. So the first step is size fractionation, filtration through 0.2 micron filter to give us our dissolved fraction. That dissolved fraction then goes through density purification, where we add it onto a sucrose cushion and ultracentrifuge. This basically lets us purify the phage from any other particles that might be in the sample. We then resuspend that phage pellet into a sterile buffer, and we put it through dialysis in a sample, or sorry, in a water bath, which we change many, many times. This basically gets rid of any unincorporated labeled iron. So when we're left with our final sample, it should just be purified phage, and we can see if they've incorporated the label. So here's the data from one of the trials, and it's a little bit of a busy graph, so I'm going to walk you through it. On the y-axis, you have the iron 57, the amount of iron 57, and then along the x-axis, you have your various steps of purification. So first, the cell culture. Green is incubated with phage T4, which is the one that we know has iron. Blue, T5, which we don't know. Red is just the bacterial control with no viruses, and yellow is a blank. And you can see that everybody starts with high iron 57, but as we go through lysis and we start doing our purification through filtration, then our ultracentrifugation and dialysis, these concentrations of iron really drop off as we're purifying the viruses. Now I'm going to zoom in on this red box here because compared to that, it looks like there's nothing there, but this is where the really interesting data are. And what we see, here's the same graph I showed you, and here's the zoomed in red part, is that after we do our final step, so dialysis purified, here's our final phage fractions, we see significantly more iron 57 associated with phage T4 and phage T5 compared to bacterial controls that didn't have any viruses. And Chelsea repeated this through five independent trials. Each had 3 independent replicates within it. And I'm not going to show you all the data today for the sake of time, but I will show you kind of the end result. So we then looked at how many viruses were in these purified fractions at the end. And so it doesn't look quite like this because remember the bacteria aren't there, but we used the same method to count the viruses. And then we took our concentrations of iron, or sorry, our amount of iron, and we divided by the number of viruses to calculate the number of iron 57 atoms per phage. Round 1 only had T4, the rest of the rounds had T4 and T5. And you can see, even though there's some variation within the numbers, which we actually believe to be due to inaccuracies within the accounts, we see somewhere in the hundreds to thousands of iron 57 atoms being incorporated per phage. which I think is actually a pretty narrow window for an actual experimental system. Our next step on this project is to now take these ideas that we've successfully developed the methods for in E. coli and move them into a marine system. The marine system we're going to be using is called Vibrio nitrogens. It is a bacterium, it's a Vibrio. It's normally found in marine and estuarine environments. But the nice thing about this is that it's super easy to grow. Much like E. coli, it actually doubles faster than E. coli, which is something we very rarely see for marine systems. Our lab has sequenced and developed 4 different vibriophage to be able to use in these assays. You can see their EMs below, and this is work that was led by postdoc E. Wynn in our lab with 2 amazing undergrad students, Noah Jonas and Paige Bowman. So overall, phage-bound metals may be really important for many ecosystems. We're going to obviously take this into different phage host systems, both tailed and non-tailed phage, as well as different hosts and environments. We want to try to do some competition experiments between siderophores and the phage for host receptors and see if the siderophores are actually capable of binding and stealing iron from the phage tails. And finally, because of the methods that are used for quantifying the iron, the ICP-MS, we can actually examine a full suite of metals and not just iron to see if there are any other metals that phage may be important for. And finally, since our lab logo is Focus is for Losers and we work on lots of different things, I'm just going to leave you with these photos of our current lab members and a brief description of each of their projects since I couldn't talk about everything today, and I'd be happy to take any questions.
Thanks, Maya. This is great. Any questions from students first? I hear echo. Please mute your microphone.
Catherine.
Go ahead.
Hi, I'm gonna try this with the screen. Oh, hi Maya. Great talk. So I'm only asking this question because of Tim's presentation. Have you thought about doing iron isotopes at all, or if that could help you answer any questions?
Yeah, that's a great question. And Tim, feel free to chime in. Right now, while we're doing the lab experiments, we're kind of artificially adding an isotope, right, just so that we can be able to trace it. It gets a lot trickier if we start just using natural abundance iron, because it turns out everything we do as microbiologists, like autoclaving in a giant metal casket, introduces iron contamination. I'm not sure. I think it's a super fascinating idea, but I'm not sure how we would do it. I would like to look at whether there's active fractionation that happens during infection processes, and that might be a way to get at that.
Yeah, I'm impressed. I saw iron 57 as your isotope. I'm just impressed that you're able to measure iron at low enough levels. We deal with contamination a lot.
Yeah, I think the isotope has been our savior on that front. Brianna.
Hi. So I was wondering, when it comes to, so if I understood correctly, you were saying that it seems like a lot of, if not most of the iron that's kind of in the water column is going to be bound up to viruses. And so obviously that's not the same thing as being dissolved, but for the purposes of, say, like phytoplankton, is it like do they have free access to iron that's bound up in viruses because viruses are so much smaller than phytoplankton are, or is that substantially different in terms of like what's bioavailable to photosynthesizers?
Yeah, it's a great question. So I think our general idea is that this is probably not bioavailable because it's bound up in viruses. But we don't really know that. We've done some kind of brief modeling studies with Leon Hardy at USF St. Pete to just kind of try to figure out how tightly that iron is within the tails, because you notice I used the word that it was like coordinated by those histidine motifs, and I'm a little antsy to say bound because we don't know how tight that interaction is, or whether it would be accessible by something else. And so that's where I think we really need to do these competition experiments. From what I understand, iron binding ligands is a whole wide, diverse pool of things. And so viruses would only be one part of that, but it's a part I'm hoping we can get a handle on. We're also interested in knowing how, when viral particles decay, what happens to that iron, right? Or even when a virus infects a cell, its tail doesn't actually end up inside the cell, as far as we know. It injects its DNA, but we don't know if the cell actually can access that iron or if it gets released. All good questions.
Thank you.
Any more questions?
I will turn it over to Ali.
Yes, please. So next, we're going to have Dr. Ali Graham, who is a geological oceanographer in this college, relatively new. He's going to talk about the tides and the glaciers. Ali, please, go ahead. Please unmute yourself.
Hello, can you hear me now?
Yes.
Great.
I hear you.
I ran ahead of myself. Okay, can you guys see that okay?
Yes.
Awesome. Okay, thanks, Shonmin, for the introduction. Maya said in her talk, focus is for losers. I'm afraid I'm going to have quite a focused talk today. I've chosen to hone in on just one piece of work I've been doing kind of recently since arriving at CMS. But I think it's a good one because it showcases or gives you a flavour maybe for what makes me tick as a scientist and what floats my boat, what I'm working on, and hopefully kind of the topics and themes that my lab and I will be focusing on in the coming kind of years. So I should say now that I do have a lab, which is a good news as well. It's been a while getting it refurbed, but it's now almost ready to go. It's downstairs in MSL. Obviously, COVID's put a bit of a stopper on me getting in there and kitting it out, but I'm working now on bringing in some sort of fresh faces and some bright minds in the next year or two. So kind of watch out for, I guess, the group building slowly but surely. So what I want to talk to you today about is some work I've been doing based off a cruise from last year, 2019, the start of 2019, looking at this historical retreat of Thwaites Glacier in West Antarctica. I'm really focusing in on just one tiny study that forms part of a larger project that's using an autonomous underwater vehicle, this orange submarine that you can basically see in the title slide here. So for the wider context of this, I'm a co-PI on a project that's called THOR. It stands for Thwaites Offshore Research. And it's a 5-year NSF NERC funded project that's looking at Thwaites Glacier from an offshore perspective from the marine realm. And this sits under kind of a flagship, a wider program called the International Thwaites Glacier Collaboration that's joint UK-US, as well as a whole of other a load of people from different nations. And really the goal of this program over the next 5 years is to try and look at how Thwaites is changing in the future. So we sit under this flagship program, and we're doing our own bits of work that are contributing to it. There's a whole bunch of co-authors that I should acknowledge, but not least Anna Varlin, who is becoming a close collaborator of mine at the University of Gothenburg in Sweden. She was instrumental in acquiring the sub that you can see on the screen, testing it and trialing it, making it sea ready and polar sea ready, and then allowing me time to basically work with it and mess around with it at the seabed. So what I'm focusing on today is just the data from this one sub, from one mission, and I'm going to be kind of honing in on some interesting features that tell us something about retreat at Thwaites. So if you don't go away with anything else today, it's probably I want you to take home this.
Ali,
Argue that
we
it's not the
lost
most
your...
important. It's a huge glacier. It's the map on the left is from Eric Rigneau's work in 2011. It shows velocities for the Antarctic ice sheet. And I'm drawing on here the drainage basins of two kind of key catchments, and Thwaites is drawn on marked with a T. The size of that drainage basin is equivalent to the area of Florida, so it's a big glacier. And if you took that whole blob of ice and threw it into the ocean right now, you'd raise global sea level by around 1 meter. In reality, if you did that, then you'd also destabilize adjacent basins. So the area marked P is Pine Island Glacier, it's kind of sister glacier, and also the glaciers that drain into the Ross Sea. These would all be drawn down into the basin. You'd end up with basically a partial or complete collapse of the West Antarctic ice sheet. And we're looking at then at around 3.5 meters of sea level rise from such a collapse. So Thwaites is the jugular of Antarctica, it really is, and that's why it's been the focus of this program. Right now, it's giving out a modest 0.1 millimeter per year to sea level. Doesn't sound a lot, but that's a millimeter over a decade. And what we've been seeing is that the rate of discharge into the ocean has doubled since the 1990s. And you can see on the graphic on the right-hand side, which I hope is working for you guys. I wanted to put this in just as to emphasize that the front of Thwaites is really dependent on its floating ice shelf. So where the glacier comes off its bed into the ocean, it floats away, and then it carves into these icebergs, and it delivers meltwater to the ocean. And that's the way it's churning out mass to the ocean. There's been a couple of studies, actually sort of 5 years ago now, which has suggested that Thwaites is close to, or may already have passed, a tipping point whereby the glacier will undergo a runaway retreat that is irreversible. And this has been the driver for the whole program. So really what the burning question for Thwaites is, how will it evolve in the next 200 years? It's a kind of a critical question that we need to answer, and we are contributing to it in through my lab, my project. So I mentioned that we're interested in the marine realm, coming from the ocean, looking at Thwaites, but we're also interested in the paleo, so what's gone on in the past. So what we know about Thwaites right now is actually relatively limited. We've had an observational window where we've been able to see what's been happening in the glacier from mainly using satellites, but some kind of in field and airborne programs too. And you can use some of the data there to kind of make predictions. So this is kind of the bit we've observed. This is just a schematic that shows sort of time on the x-axis and a relative ice sheet change on the y-axis. This is the bit we kind of know. It's a relatively thin sliver of time. And then this is the kind of the bit we want to know for the future. So, is there a best case scenario where maybe some self-stabilizing feedbacks kick in with the solid earth or the ocean, which stops Thwaites from collapsing, or are we on some sort of trajectory in the next century or so where the ice sheet basically loses all its mass? To get to those predictions, we need to know something about what happened in the past, because the future trajectory of Thwaites is basically a function of its past imbalances, and we know there are processes that occur on longer timescales than the bit that we've been able to observe, right? So essentially we need to go back in time, and we need to know what's gone on in the past in order to make some good predictions for the future. So we are really steering ice sheet models with the past. So my work is really, especially for this aspect of the project, looking at areas close to Thwaites and trying to address two things. One is what are the rates of change in the glacier been in the past? We know a bit about what's happening there right now. What has been the kind of the norm back in time, in historical times, and also what are the mechanisms that might lead the glacier to unpin? So the map on the left here shows you Thwaites. The hatched area at the bottom of the graphic is the area of grounded glaciers. That's where the ice is too thick to float off its bed. It's sat on bedrock. And then the black line that runs through the center of the graphic is the ice shelf edge. So everything between those is currently floating ice shelf. And I've also drawn on some red and orange and yellow lines here that mark former granuline positions. And what you can see is there's sort of two areas where the glacier is grounded. One is obviously the grounding zone right now, but also out at the terminus of the ice shelves, there's the Thwaites Glacier Tongue and the Eastern Ice Shelf, kind of are two separate limbs of the ice shelf. And the Eastern Ice Shelf is pinned now on a seafloor high, and the Thwaites Glacier Tongue has been up until recently pinned on a seabed high as well. So the future evolution of Thwaites is actually a hypothesis that some of the recent increases in discharge from Thwaites are a result of what's going on out here in the terminus, where perhaps the ice shelf is lifting off some of its seabed highs, and these are effectively a handbrake on ice discharge to the ocean. So if you lift the ice shelf off of its seafloor highs, it's going to start retreating quicker, because it's giving ice quicker to the ocean. So there's two objectives that my work here is trying to address: what were the rates of class change, and then also how do glaciers such as Thwaites unpin from these sorts of seafloor highs, because I think the future of Thwaites can be really critically linked to how these two environments evolve. So what I want to focus right in on now is an area of the seafloor just at the apex of the Thwaites Glacier Tongue, and an area we've named the ice melange shelf. It's an unnamed part of the ice shelf, but it used to be a contiguous ice shelf, and it's now breaking up into an ice melange. So kind of a whole mishmash of bergs and sea ice. And I'm going to just focus in on this area that's marked by a white box with the letter C in, and that's where we're going to be working for the rest of this talk. So what we did here was deploy RAN, the Gothenburg Kongsberg-Huygens AUV. This is it in the bottom right hand corner. And we mapped with shipborne, echo sounders, the seafloor in this area, and it's a really nice seafloor map. I'm not going to go much into it right now, but essentially there's a trough here. It's about 800 meters deep, and then in the center of this trough is something we called the bump, for want of a better word. It looks like a big bump, and this is a really nice little feature because it has the potential to be a former pinning point. And this yellow line here is the 2019 ice front. So we were working as close to the ice front as a ship could possibly go. So we deployed the AUV in this location. We ran a 20-hour mission. We undertook multi-beam bathymetry mapping, so high-resolution seafloor mapping. We collected side-scan sonar and also sub-bottom profile data, as well as a whole load of oceanographic underway instrumentation that's going to form part of another paper that's coming out relatively recently. So this is in quite deep water, 650 meters in this location, and the AUV flew at about 50 to 90 meters off the seabed. So we were able to get really high resolution images of the seafloor. So what I'm going to do is jump straight into the data. I'm going to look at the new data we collected on top of the bump, then some of the side scan. I'm going to hone right in on some of the features from the side scan, and then I'm going to show you some of the analysis I've done on and what I think it means for Thwaites, and that's going to basically be the rest of this talk. So let's jump to the top of the bump, and this is what you see when you put the data into a 3D viewer after a lot of cleaning, thanks to some of the Kongsberg guys. And what I want to point out here, well, firstly, the resolution of this data is just incredible. This is a 70 centimeter grid, so each cell in the grid is 70 by 70 centimeters, so it's sub-meter resolution. You can see several things going on here for any keen geologists watching. I'm sure there's a few. You can see the bump, and it's got mini bumps around it. Are bedrock cord. This is jointed, heavily jointed, and in some cases faulted terrain. So this is raw bedrock sticking out of the ground. This is probably granitoid. It's definitely crystalline basement. And then the tops of the bump and this kind of this edge of the bump too are markedly different. They are smooth and streamlined, and we think these are sedimented. So there's sediments covering the bedrock on the top, and they've been planed flat, and you can see there are these linear features, these lineations that are the sole print, essentially the footprint of Thwaites, where it's actually sat on this bump in the past. And you can see actually in the far field as well that there is evidence that Thwaites has been grounded through this whole region in the past, which is not unsurprising. But what we were really keen on looking at is actually some of the features on top of this bump. How has the ice left this bump in the recent past? If you go to the side scan and look at some of the top, actually I'm going to go and show you a map first. So what you can do is take the, you can not make nice pictures of them, but you can also do some detailed mapping, and that's what I've been doing for at least parts of the parts I've arrived at CMS. There's a lot going on here, and I don't want you to focus on all the detail. Essentially, the key things to take away are that what we have here as a collective is something that looks to a glacial geologist very much like a grounding zone environment that you find in any kind of relict offshore margin. And so the green lines are former grounding lines, so this is where the glacier once sat. The orange zone is the one we're really interested in, because this is the most recent surface that's been exposed by the glacier. And the green line marks out where the glacier used to sit. So it's sat on the edge of the bump, and then behind it there's a nice sedimentary surface that has sole marks on it that are these grey lines that are oriented in the direction of ice flow. And then superimposed on those, I hope you can see, these are these black squiggles. There are lots and lots and lots of these black squiggles. They are features that we're calling ribs, and they're oriented transverse, so 90 degrees to the direction of ice flow. And these features are formed since ice retreated or during ice retreat. That's a clear signal that we're getting. And you can see these even better then if you go into the side scan. You can see them on the multi-beam. But these are what they look like in side scan. So the side scan on the AV is basically giving you a measure of backscatter intensity. So it's picking out things that are highly reflective or things that are quite absorbent. And that shows you something that's rough or smooth, rock versus sediment, and it tells you something about the heights of features. So it gives you a really nice high-resolution view of the seafloor. So these are these ribs, and they are everywhere, and there are loads of them. When you first start eyeballing them in, they are maybe 6 to 7 meters wide. You can trace them. They have these kind of wavy views. If you look at the bottom left side scan image, you can trace them across multiple lineations. These lineations are these things going underneath, left to right in the image, and these are the ribs going across. In some cases, these ribs are actually oblique to the lineations, so they're not actually formed along the tracks. So it tells us something maybe about how they've formed. And in this main image on the right-hand side, what I've done here is circle with these red circles the tops of what I believe to be this is a lineation, a till ridge essentially, that runs left to right across this image. And what the ribs are doing here is essentially the process that's formed the ribs has obliterated the lineation, so the underlying signature of the glacier grounding on the seafloor, and has left these blobs, these remnant structures that line up and are basically part of what used to be a lineation. So these things, this tells us these are younger than the grounded signature of Thwaites, and we believe these formed in a retreat direction in series in a landward direction. So I was interested in how these form and what they tell us about historical retreat. We've not really seen anything like them before, although there is some work coming out now from other parts of Antarctica that are showing similar features, and have got these ideas about how they form. So what I did was looked for the longest series of these ribs that I could find, and actually they're from this area that's sort of shaped like a V. I took a profile through this long series and actually composited two together to make a series of topography that runs in the direction of ice retreat and in the direction of ice flow. So this is the bathymetry, just raw. What you can see is that the ribs on here are 10 to 70 centimeters high. They vary. They're sharp crested, at least in this view, but there's a big vertical exaggeration on this. They are spaced anywhere from a couple of meters to about 11 meters apart, and in this series we have about 166 ridges that we could analyze. So I took those data and then I've actually done a lot of analysis on their geometries and their metrics. So this is the same data set now where I've extracted the amplitude, so the heights of each of those ridges in a series going from north to south. So left to right is north to south in the direction of what we believe to be ice retreat. And what you pick out really nicely here, we've got amplitudes and in meters on the y-axis, just numbered landforms in series on the x. You can start seeing there's this cyclicity, especially in the center part of the profiles, and this cyclicity seems to fall around 13 to 15 ridges in every cycle. You can look at the spacings of the ridges too, and what you find is that the two parameters co-vary, so that the cyclicity in the heights is also mirrored in the spacing. And what this is basically telling you is that when ribs are farther apart, they are also taller. So there's a mechanism that's linking the spacing and the heights together. And we seem to have about 10 cycles, at least well preserved here, potentially a few more. What becomes then very interesting, and where tides start coming in, is that obviously this 13 to 15 cyclicity started making me think about the number 14, which in terms of tides is a very significant number because spring neap cycles I know are dominant in other parts of Antarctica where we've been able to observe the motion of ice shelves. So what I've done here is extract a 6-month time series of tidal predictions from the Oregon State University TDX09 tide model. You can do the same with the CATS08 model for Antarctica, which is Laurie Pavlin's model, and you basically find the same thing. In grey here in the back, this is the tide model. It has a strong diurnal signal, so there's one main high water and one low water per day, and you can see there's a fortnightly, the spring neap constituent comes out really nicely in the tidal model as well. What then is really cool is if you line these things up and scale the tide model so that one rib equates to one tidal day, then the data and the tide model seem to strongly correlate, especially in these kind of center 4 or 5 cycles. So tides and landforms seem to line up really well, and this is a tide model in time series. So what we're saying essentially is that what we seem to be showing is that the ribs form as a time series. They form one per day, and they form in a landward direction. The implication here then is that this blue line, which is the spacing, is essentially showing a daily rate of retreat of the glacier through time over 5.5 months. So we've got these triangles are essentially monthly intervals. So at some point in time, we've actually been able to pick out the retreat cycle, the retreat pattern of Thwaites back in time over almost half a year. So tides definitely modulate the landforms, and I've actually said the stuff on the right-hand side here, so you can ignore that. I've done some spectral analysis that basically confirms what we suspect from eyeballing this, that the heights of the ribs, the spacing of the ribs, as well as the tide model all share really nice peaks in their frequency spectra, and so this is the 14-day cyclicity, the spring neap cycle. It comes out really nicely in all 3 sets of data. So tides seem to be really linked to the landforms and are modulating how they are being created, and that's led us then to hypothesize and model how these things are created. So this is a 3-step model of formation. At the bottom here, you can see a typical tidal cycle at Thwaites Glacier with a diurnal signal. We've got this strong high water, strong low water, and a very muted sort of semi-diurnal signal that's on the background here. So in step 1, effectively the ice is settled on its sediment bed, and through extrusion is pushing up a small ridge at the grounding zone. So these things we believe are forming right at the grounding zone. 10 to 70 centimeters high, which is what we've been observing. During the high water, the main high water of the day, you draw the ice shelf base upwards and the grounding line migrates backwards. What this does is actually open up a small cavity above the rib, and we think the ocean then plays a role too here. So through tidal mixing, you actually draw in warm water, relatively warm water, that is then enhancing melt the underside of the ice shelf, and that melt is actually then driving a persistent retreat backwards through time. So we think the ocean is playing a big role, and tides are kind of influencing how the ocean is delivering heat to the grounding line. And these, what I should say is that the grounding zone is a very poorly prioritized and understood place for ice-ocean interactions. We don't know a lot about what's going on there, but there is work from the neighboring Getz ice shelf, from Anna and her team, where they deployed instrumentation through the ice shelf, and they seem to show that basal melt rates, so how much is melting at the base of the ice shelf, follow a very strong spring leap cycle. So this kind of model fits very well with some observations that we're making from other ice shelves in Antarctica. And then at the third phase, essentially the next low water, the ice shelf settles back down in its new position, and it extrudes another rib in series. And there is if anyone's interested, there is a paper out in Science by Julian Dowdeswell and his team that have proposed a somewhat similar model for ancient retreats of glaciers on another part of Antarctica, which haven't done any of the kind of analysis that we've done here. So what are the implications? Our data, I think, is showing more than 5 months of retreats in Thwaites Glacier in the past. If you do a kind of time averaging of the data set, the spacing of those ribs, what you find is the retreat rate seems to be very high. It's around 2.2 to 2.3 kilometers per year. If you look on the map on the left, this is from Thwaites, although it's a bit hard to orientate yourself here, but take my word for it. These two numbers here, 0.6 and 0.8 kilometers per year, is the average ice retreat or granuline retreat rate for the time period 2011-2017. Over in the east side over here, so this is actually east because it's flipped around, but this is the largest measured granuline retreat rate for the whole of Thwaites, which is 1.2 kilometers per year over those 6 years. So we seem to be finding retreat rates in some sort of distant past that are, at least twice, but maybe 3 or 4 times the rate of what we believe is modern retreat rates at Thwaites. If you compare these then to ancient rates of granular retreat that have been reconstructed from satellites or airborne geophysics, typically, we're looking at retreat rates on the order of less than 1 kilometer per year over decades for glaciers like Thwaites. And in offshore geological records, where you look over millennia, the retreat rates are even lower. So I think an implication is that what we're finding in a very high resolution window at Thwaites is that there are pulses of rapid change that maybe aren't picked up by these other methods that are trying to reconstruct past granular retreats. I think there's something also to say about mechanisms of retreat. One thing we were faced with when trying to explain this model is how you actually flex a grounding line that's is fixed to an ice bed. And one way of doing this is to invoke something that is a kind of neat little glaciological quirk, which is called an ice plane. This graphic from the right-hand side is quite old now, but it comes from Hugh Corr's work at Pine Island Glacier. And essentially, ice planes are regions of a grounded glacier where the region upstream of the grounding line, so where it's fixed, so the bed is still near buoyant and actually can still flex, and it's very lightly grounded, so that the ice surface is only a few meters or tens of meters above buoyancy. So this is a nice kind of way of explaining, I think, how the ribs formed in the past. You only need small amounts of thinning at the base here in order to reach buoyancy and to peel back an ice plane from its bed. So only small amounts of thinning need to be invoked in order to instigate quite fast, non-linear rates of retreat. And that's what we believe we've picked up or observing in the C4 record at Thwaites. I guess then the last question is, when did these things form? And that's what a lot of people have asked me so far when I've shown them. These the graphic here shows the area in pink is the area where we've collected the data and where we have ribs formed on top of this high, and the modern granular line is about 55 kilometers to the south. We know that from through ice radar observations that the granular line was already south of this point in 1978, so it predates 1978. If you use a modern granular line retreat rate to kind of project to where we are now, or project out to the study site, then you can maybe put yourself in the 1950s when these things formed, when the glacier was out here. If you take a more modest rate, which I think is probably the correct way to go, a more conservative rate, then maybe 180, 200 years ago was when the glacier last sat out here. But I think either way you look at it, these ribs formed in an area that predates satellite observations and provides us with a super high resolution image of what the glacier's been doing in the past that we've not been able to observe before. And there's actually no way of, even in the modern day setting, of being able to understand retreat rates at this kind of daily resolution. It's quite remarkable we've been able to do it with seafloor data. So the take-home messages for today, essentially, that rates of retreat at Thwaites were faster, at least twice, but maybe 3 or 4 times faster in the pre-satellite history of Thwaites than is recorded in the period 2011 to 2017. You can sort of draw your own conclusions right now on implications about what that might mean. We're still working on this and writing this up, but it's quite stunning that we found something that seems to show really fast pulses of retreat in the past. I haven't really focused in on this, but the geometry of the bed and the way the ice base is shaped is probably to some extent preconditioning the glacier to rapid retreat. So the fact that you've got a very flat sea floor with a low slope is needed in order to generate an ice plain. So it might be a combination of ice and bed and tidal factors that come together in certain scenarios to make these kind of pulses of fast retreat occur. What we can say, I think, is that you don't necessarily need to have a rapid change in external forcing in order to trigger some of these events. You can have just a small amount of basal thinning, a glacier like Thwaites, and you can suddenly trigger quite a rapid peel back of the granular line. So you don't necessarily need to keep warming up the ocean. Today's kind of basal mount rates might be enough to trigger faster events in the future. And actually, the final point here is essentially that fast non-linear pulses of ice recession probably characterized the way big glaciers like Thwaites unpinned from its seafloor high in the past, and probably will do so in future. So I think it wouldn't be surprising if in the coming 50, 60, 70 years that we suddenly see rapid changes in parts of the granular line where it's sat on seafloor ridges. Okay, and thank you for listening. There's a lot more from Thwaites that I would like to tell you at some point from this season that we've just undertaken, but I'm happy to take questions and or receive emails and things offline. Thanks.
Thank you, Ali. We are a few minutes behind schedule, but because this is the last talk, we have a few minutes for questions.
Sorry for running over a little bit. I just got carried away.
Yeah, if you have questions, this is a good time.
Everyone's keen for beers. That must be it.
Yeah, I can't see raised hands or any symbol. Renate, perhaps you can see.
No, it doesn't look like there's anyone with questions.
Okay.
Oh, there's one, Brian O'Malley.
Brian. Hey, Brian.
Hey, Dr. Graham, how are you? That was a great
Hey.
presentation. Thank you. So I was wondering if you think that this, can be preserved in the sediment record as pulses as the glacier melts. I'm sure you have some kind of sediment trapped in that ice, right? And over time, that's going to eventually, deposit. And I was wondering if maybe you could track this in the more modern era with radioisotopes. Like as you get closer to today's glacier, you would see the lead-210 spike getting shallower and shallower in the sediment record as you approach the glacier. So I was wondering if you guys, if you even took core samples on your, last trek, or if it was all kind of multi-beam stuff.
Okay, good thinking, good question. We unfortunately didn't sample the bump, the area around where the AUV mission was, and that was largely because of the time constraints we had in the region. Ice was closing in, and then we revisited this year with a grand hope that we would be able to go back to those sites, but we never got within 30-40 kilometers of those sites again. The ICI situation was just so bad. So we don't have samples that can kind of ground truth any of those ribs and the things I've shown today. We do have lots of cores from other parts of the seafloor around Thwaites, and there are teams working on the lead-210 records from those cores, primarily to date to see what the sedimentation rate is in the upper kind of sequences. But yeah, there will be an interesting lead-210 story, I think, coming out of Thwaites, and about where sediments are deposited and at what rates as well.
Yeah, definitely. Thank you.
David Nara, your hand is up.
Yes, I'll put it down. So I had two quick questions. Well, maybe they're not that quick, but obviously trying to core the sediments underneath the glaciers would be of great interest. And I'm just curious regarding technology. Will we get to a point, do you think, that we can get AUVs to go underneath the glaciers and core in certain strategic areas that the ships won't want to go to? And my second question is, in your talk, you mentioned we may be at the point of a runaway glacial sliding. When do we know for sure? What is going to be the criteria that lets us know when the glacier is it's out of control type of thing?
Yeah, good question. Let me deal with that one first, because that's probably that is a pressing question. We debated this on the ship. In fact, standing there, looking at Thwaites, talking to one another about if Thwaites was collapsing, what would that look like? And even with us looking at it, with our naked eyes, are we observing it? And I don't know if anyone's actually really answered that question. It's like, over geological timescales, we can observe collapse happening, but nobody's ever seen an ice stream or a marine ice stream collapse over a kind of human lifetime. So the signature of what it would look like is tricky. And I guess what our project is doing is trying to kind of, in some parts of it, trying to assess in some ways, has there been a change in the recent past that is a signal, like a smoking gun, that something has changed? So is there more meltwater coming out? Have the sedimentation rates drastically changed? What we seem to be finding in these ribs, at least, is that the retreat rates recorded by the landforms are, even faster than what the retreat rate is right now, at least what we believe the retreat rate is, which is sort of counterintuitive, but it suggests that there has been pulses of fast retreat that are as fast, or if not quicker, than what we're observing with satellites there today. I don't know. I guess that doesn't really answer the question. I think we don't really know what the signature will be. I think ice sheet models will basically give us, when we've been able to constrain them well enough to be confident about the projections, they're the ones that will tell us what kind of timescale we're looking at to retreat the glacier back into its basin, if it will retreat back in there at all, but we suspect it will. And that whether that's 100 years, 200 years, 1,000 years, we're not sure yet. The other question was about AVs going underneath glaciers. So we are already doing that. In fact, this mission to the bump did go very small way, just a tiny bit underneath the floating ice edge. I've been involved with work that has sent another, the Autosub 3 back at Pine Island Glacier. We sent that underneath Pine Island, a quite considerable way. That went under about 30 kilometers in and out to the cavity. we've not yet got a way of coring from an unmanned platform. There are ways potentially of doing it with an ROV, with a light tether, and that's something I've been talking to Woods Hole about. Maybe in future they've got something that could fit the bill. One of my colleagues has been taking an alternative approach, which is going on to the top of the ice shelf, drilling a hot water borehole, and then deploying a gravity core through the hole before it freezes up. And that does actually work. That you get yourself some nice sediment cores that way, but you're coring blind basically through an ice shelf, which is not the best approach. So there are ways of doing it, and we're getting better with technology. There's something called IceFin, which if you guys all come to Brittany Schmidt's seminar in I don't know when that is, maybe next month or the month after, she's at Georgia Tech, and she has an amazing thing called IceFin, which they can deploy through. It's an AUV deployed through an ice borehole, and it does some amazing stuff, and you can get it to places that no other person or vehicle has got to before. So watch out for that. That's a good plug for her seminar. But yeah, thanks for the questions, Dave.
Yeah, I should point out that Woods Hole does have a pretty creative group. They put a pogo stick on the autonomous benthic explorer, ABE for short, and they were able to sample glass from lava flows by this waxy tip protrusion that would go down and break the glass and capture glass off of lava flows. Coring sediments, of course, would be much more difficult, but at least they're on the right track.
Yeah, I'm all for these. For if it works, then do it. I mean, some of the coring done through the ice shelf uses a skidoo basically to as a percussion corer. So they just hammer in the corer with a skidoo. I mean, it's rough and ready, but it works. So
Ryan just sent a link for a Georgia Tech AUV.
Yes, excellent. Thanks, Ryan. I guess, Ryan, you might be the host or when it comes to it. So contact Ryan. I'm certainly going to be there as well at that meeting. So
see more questions now? Renate, do you see more hands?
Yes, Nancy Williams.
Yeah. Hi, Ali. Great talk. Hi. So I was wondering, I guess, so it sounds like this method for estimating the grounding line retreat, you don't have any satellite data from that period, so you can't really do an intercomparison. But the uncertainty on that seems pretty small. So, if it is tidal, then, you have you're pretty certain in that rate. How uncertain are the other ways of estimating? What are the uncertainties on the satellite-based?
So the satellite based ways of measuring granular line retreat are essentially taking probably individual snapshots in time from a given year or season, and then looking at how it's changed in the following one, if you're lucky, or maybe even several years later. I know for fractal weights, there's been time periods in the last sort of 30, 40 years where there's a whole decade or so missing. So what we're just doing is taking a broad look at how has the grounding line changed over a few years at a time, and just fixing a number on where it was at one point and where it is now. So even those kind of measurements might be hiding a lot of variability in the way the grounding line's behaving. Grounding line retreat doesn't, as a measure, a kilometer per year might hide readvances in the grounding line or variations in the position from 1 week to the next. So I wouldn't be surprised if there's a lot more variability that we're not picking up, even in the way we're observing the ice sheet today. And there's probably a challenge out there for somebody to be able to monitor grounding line changes at a much higher resolution. It's not me, but somebody else, a glaciologist. So, yeah, good question.
Yeah, we are way over schedule. If anybody has a question, we may address the last question.
It doesn't look like anyone has any.
Okay. If so, thanks, Ali. Yeah,
Thanks, Roman. Thank you, guys.
Thank you. Well, this is a great turnout. I watched the number of participants all day. On average, we have a 70, and this is way more than the in-person seminar. This is great. I want to thank all speakers and thank the audience, and particularly thank Renate for organizing everything. So next week, we'll start our weekly seminar series hosted by Amelia. I hope to see you there. So if there's no comment, question, I think we'll end today's seminar. And again, thank you all. This is a long day. I hope everyone learned a lot. I did. And if you have more questions, you know where to find the speakers, right? And not physically, but you know their emails now. Thank you. You all have a great weekend. Stay safe.
Thank you.
Bye-bye.